Cellular respiration in bacteria, mechanisms and adaptations. Cellular respiration in bacteria, mechanisms and adaptations. Introduction. Cellular respiration is a vital metabolic process that occurs in all living organisms, including bacteria. It is the process by which cells convert nutrients into energy, specifically adenosine triph phosphate, ATP. Bacteria have evolved various mechanisms and adaptations to carry out cellular respiration efficiently in diverse environments. This article aims to delve into the mechanisms and adaptations of cellular respiration in bacteria. Overview of cellular respiration. Cellular respiration consists of three main stages: glycolysis, the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, and oxidative phosphorylation. These stages are within different parts of the bacterial cell, including the cytoplasm and the cell membrane. Glycolysis. Glycolysis is the first step of cellular respiration and takes place in the cytoplasm of bacteria. It is a universal pathway that occurs in both aerobic and anaerobic organisms. During glycolysis, glucose molecules are broken down into two molecules of pyruvate, generating a small amount of ATP and reducing equivalents, such as NAD. Krebs cycle. The Krebs cycle, which occurs in the bacterial cytoplasm or mitochondria-like structures, further oxidizes the pyruvate produced during glycolysis. In this cycle, the remaining carbon atoms from the original glucose molecule are oxidized, releasing more reducing equivalents and ATP. The end products of the Krebs cycle include carbon dioxide, CO2, and reduced coenzymes, such as NAD and FOD2. Oxidative phosphorylation. The final stage of cellular respiration, oxidative phosphorylation, occurs in the cell membrane of bacteria. This stage utilizes the reducing equivalents generated during glycolysis and the Krebs cycle to generate a large amount of ATP. The process involves the transfer of electrons through the electron transport chain, etc., and the synthesis of ATP through chemiosmosis. Mechanisms and adaptations in bacterial cellular respiration. 1. Oxygen requirements and adaptations. Some bacteria are obligate anaerobes, meaning they can only survive in the absence of oxygen. These organisms have adapted alternative mechanisms to carry out cellular respiration, such as using nitre or sulfate as electron acceptors in anaerobic respiration. On the other hand, facultative anaerobes can switch between using oxygen and alternative electron acceptors depending on the availability of oxygen in their environment. 2. Diverse electron transport chains. Bacteria have evolved various types of electron transport chains, EECs, to accommodate different environmental conditions. For instance, photoautotrophic bacteria utilize light energy to drive their EECs, while chemolithotrophic bacteria use inorganic compounds as energy sources. Bacteria that live in extreme environments, such as acidic or alkaline conditions, have specialized adaptations in air EECs to survive these harsh conditions. 3. Aerobic versus anaerobic respiration. Aerobic respiration in bacteria utilizes oxygen as the final electron acceptor, producing water as a byproduct. This process yields more ATP compared to anaerobic respiration. Anaerobic respiration, on the other hand, uses alternative electron acceptors such as nitrate, sulfate, or carbonate. Bacteria that carry out anaerobic respiration have adapted enzymes and transport systems to utilize alternative electron acceptors effectively. 4. Energy conservation mechanisms. Bacteria employ different energy conservation mechanisms based on their metabolic requirements. For example, some bacteria can generate ATP solely through substrate-level phosphorylation during glycolysis and the Krebs cycle. Others rely more heavily on oxidative phosphorylation to generate ATP using the proton motive force generated across the cell membrane. Cellular respiration in bacteria is a highly efficient process that allows these microorganisms to convert nutrients into energy. The process consists of three main stages: glycolysis, the Krebs cycle, and oxidative phosphorylation. Each stage occurs in different parts of the bacterial cell, such as the cytoplasm and the cell membrane. During glycolysis, glucose molecules are broken down into pyruvate, generating a small amount of ATP and reducing equivalents. This process takes place in the cytoplasm and is a universal pathway in both aerobic and anaerobic bacteria. The pyruvate produced in glycolysis then enters the Krebs cycle. This cycle further oxidizes the pyruvate, releasing more reducing equivalents and ATP. The end products of the Krebs cycle include carbon dioxide and reduced coenzymes. The final stage, oxidative phosphorylation, occurs in the cell membrane. It utilizes the reducing equivalents generated in glycolysis and the Krebs cycle to generate a large amount of ATP. This process involves the transfer of electrons through the electron transport chain and the synthesis of ATP through chemiosmosis. Bacteria have developed various mechanisms and adaptations to carry out cellular respiration in different environments. Some bacteria are obligate anaerobes, meaning they can only survive in the absence of oxygen. These organisms have adapted alternative mechanisms, such as using nitrate or sulfate as electron acceptors in anaerobic respiration. Facultative anaerobes can switch between using oxygen and alternative electron acceptors depending on the availability of oxygen in their environment. This adaptability allows them to survive in diverse conditions. Bacteria have also evolved different types of electron transport chains to accommodate various environmental conditions. Photoautotrophic bacteria use light energy, while chemolithotrophic bacteria utilize inorganic compounds as energy sources. Bacteria living in extreme environments have specialized adaptations in their electron transport chains to survive harsh conditions. Aerobic respiration in bacteria uses oxygen as the final electron acceptor, producing water as a byproduct. This process yields more ATP compared to anaerobic respiration. Anaerobic bacteria use native electron acceptors, such as nitrate or sulfate. They have adapted enzymes and transport systems to effectively utilize these alternative electron acceptors. Energy conservation mechanisms in bacteria vary depending on their metabolic requirements. Some bacteria can generate ATP solely through substrate-level phosphorylation in glycolysis and the Krebs cycle. Others rely more heavily on oxidative phosphorylation, using the proton motive force generated across the cell membrane. Cellular respiration in bacteria plays a crucial role in their survival and adaptation to different environments. Bacteria have developed unique mechanisms and adaptations to efficiently carry out metabolic process. One of the key adaptations in bacterial cellular respiration is related to oxygen requirements. Some bacteria are obligate anaerobes, meaning they can only survive in the absence of oxygen. These organisms have evolved alternative mechanisms to carry out cellular respiration, such as using nitrate or sulfate as electron acceptors in anaerobic respiration. This allows them to thrive in environments where oxygen is limited or completely absent. On the other hand, facultative anaerobes have the ability to switch between using oxygen and alternative electron acceptors based on the availability of oxygen in their surroundings. This adaptive Ility enables them to survive in diverse conditions, where oxygen levels may fluctuate. Bacteria have also evolved diverse electron transport chains, EECs, to accommodate different environmental conditions. For instance, photoautotrophic bacteria utilize light energy to drive their EECs.